right, hey everyone, we're going to do a quick recap of cellular respiration. So what is cellular respiration? Cellular respiration is just the breakdown of glucose into biochemical energy called adenosine triphosphate, or ATP. And ATP is super important because it's the energy currency of the cell used in almost all cellular processes. I said almost all, by the way. It's not used in everything. There is passive transport, for instance, that doesn't use ATP, but ATP is used in active transport. It's used in molecular synthesis, and it's also used in muscle movement. So it's incredibly important for our bodies to produce this ATP for cellular processes. Cellular respiration, of course, primarily occurs in the mitochondria. There is one stage, however, that occurs in the cytoplasm that we'll talk about in a second. Cellular respiration is primarily a catabolic process because you're breaking glucose down into smaller carbon compounds. There are two types of cellular respiration. There's aerobic respiration, which uses oxygen as a final electron acceptor, and there's also anaerobic respiration that occurs in the absence of oxygen and instead uses iron, manganese, or sulfate in its reaction. So this is the formula for cellular respiration. You guys should know by now that on the left-hand side of that arrow, those are your reactants. On the right-hand side are the products. And so glucose and oxygen in aerobic respiration are your reactants. In anaerobic respiration, only glucose would be your reactant, but this formula is specifically in regards to aerobic respiration. And so you have glucose and oxygen as your reactants. Ultimately, at the end of cellular respiration, you are going to have produced carbon dioxide, water, and ATP. So, first, where does the sugar or the glucose come from? Well, humans are heterotrophs, so we're obtaining our glucose from what we eat. If you're a plant, plants make their own glucose through the process of photosynthesis, but we have to obtain our glucose from what we eat. The oxygen, of course, comes from the air that we're breathing in, and it's plants that ultimately produce oxygen as a byproduct of photosynthesis. And that oxygen we're breathing in, and we're using that oxygen in our cellular respiration. So this is cellular respiration in a nutshell. You have three stages. You have glycolysis, which occurs in the cytosol. You have the Krebs cycle and the electron transport chain and oxidative phosphorylation that occurs in the mitochondria. You'll notice that both glycolysis and the Krebs cycle are considered substrate level phosphorylation, and that is because they directly produce ATP. And you'll notice the electron transport chain is involved in oxidative phosphorylation, and all that means is that you're getting these electrons from the coenzyme molecules NADH and FADH2, and those electrons, as they go across the electron transport chain, are resulting in the movement of those hydrogen ions into the intermembrane space, and then those hydrogen ions go through ATP synthase, and that's how ATP is produced, and that is why it's called oxidative phosphorylation. And so substrate level phosphorylation is the direct production, of ATP. So these are just some of the key players in the process of cellular respiration, first of which is glucose, which is a 5-carbon molecule. You also have oxygen, which is the final electron acceptor. You have ATP, which is the fuel that we're going to create for cellular processes. You also have coenzymes, which are just non-proteins that are necessary for the functioning of an enzyme. One of these coenzymes is nicotinamide adenine dinucleotide, or NAD+, which gets reduced to NADH. These are just coenzymes, or they're also vitamin B derivatives. Another coenzyme used in this process, or made in this process, is flavin adenine dinucleotide, or FAD, which is reduced to FADH2, also a coenzyme and vitamin B derivative. And then lastly, we have ATP synthase, which is the enzyme that we're going to use to produce the majority of the ATP. So moving on to the first stage of cellular respiration, which is glycolysis, and glycolysis occurs in the cytoplasm. It is an anaerobic process, so it doesn't require oxygen to occur. And it is exactly what the name implies. It is the splitting of glucose into 2 3-carbon molecules called pyruvic acids or pyruvates. And so if we look at the picture over here, you see our molecule of glucose. In order to break it down, we have to invest ATP. We have to invest energy. And so we end up investing 2 ATP per 1 molecule of glucose. This ultimately makes this molecule into a fructose 1,6-biphosphate. This is unstable, and so it ends up splitting into DHAP and glyceraldehyde 3-phosphate. DHAP ultimately will be converted into glyceraldehyde 3-phosphate, and glyceraldehyde 3-phosphate is going to get converted into pyruvate. So ultimately, you end up with 2 pyruvate molecules per glucose molecule. Now through this process, you also get the production of 2 NADH molecules. That's 1 NADH per pyruvate molecule. And you also get the production of 4 total ATP. That's 2 ATP produced per pyruvate molecule, 4 ATP totally produced per glucose molecule, for a net production of 2 ATP. Because we invested 2 ATP, 4 minus 2 equals 2, a net production of 2 ATP. So now we're going to move into the Krebs cycle. This is where we're going to produce a majority of our coenzymes, the NADH and the FADH2. Okay, so in glycolysis we produced 2 NADH per glucose molecule, and we also produced 2 net production of ATP per glucose molecule. Now we're going to make more NADH and a couple more ATP. Before we enter this cycle though, we have to further oxidize pyruvate to become acetyl coenzyme A. So this is a process called pyruvate oxidation. So we oxidized glucose to form pyruvate. Now we're further oxidizing pyruvate to become acetyl coenzyme A. This actually ends up in the production of 2 NADH molecules. I don't have a picture of it here, but you guys should know that pyruvate oxidation results in the production of 2 NADH molecules. Now that acetyl coenzyme A is going to enter the Krebs cycle, and when it enters the cycle, it's going to react with a molecule called oxaloacetate. And once they react, they're going to form the molecule called citrate, which is where the Krebs cycle gets its alternative name, the citric acid cycle, and even the tricarboxylic acid cycle. So now we've formed citrate, and we're going to further oxidize these carbon compounds. Okay, along the cycle, you are oxidizing these carbon compounds, and you're going to get the byproduct of carbon dioxide lost in this process. It's just a waste product that we exhale. And you're also going to get the production of NADH along this cycle. And so you'll see here that you have NAD+, which is an oxidizing agent that's accepting electrons to be reduced into NADH. You also have the production of an ATP molecule. And so here in this picture, it's GTP, but GTP is just another energy-carrying molecule equivalent to ATP. So during this process, it could make either or. Okay, so it could be ATP or it could be GTP. Either way, you're only producing 1 ATP or GTP per cycle. Okay, you also get the production of 1 FADH2, and then there's another NADH. So 1 round of the Krebs cycle gives us 1 ATP molecule, 3 NADH molecules, and 1 FADH2. And then of course you have 2 carbon dioxides that are lost as byproducts. They're just waste. But that means, right, so 1 glucose is split into 2 pyruvate molecules, which means that there are 2 turns of the Krebs cycle per glucose molecule. Which means per glucose molecule, you're getting the production of 6 NADH, 2 FADH2, and 2 ATP. Okay? Now those NADHs and FADH2s are going to go to the electron transport chain. But before we get there, you guys were wondering exactly how these coenzymes are reduced. So NAD+ and FAD, again, are just oxidizing agents that are going to accept electrons from other molecules, thereby becoming reduced. And we call them reduced because their charge decreases. by accepting those electrons. So that also means that NADH and FADH2 are reducing agents that will donate their electrons. So let's look at how it happens, how it occurs, how we reduce the NAD plus molecule, and this is basically the same for FAD2. So in order to reduce the NAD plus molecule, it has to accept 2 electrons, and it is in the form of a hydrogen ion. Now I know in the discussion sheets, the way it was worded was a little weird, so hopefully this will fix it. So the only thing you have to remember is that a hydrogen ion has 2 protons and 2 electrons. And so when you add that hydrogen ion, you are effectively adding 2 protons and 2 electrons to an NADP plus molecule, therefore reducing the charge of the molecule to become NADH. And so you also see that the bonds are moving around as well. You'll notice the molecule on the NAD+, the bonds are in different locations, and in NADH, the bonds move around and you lose a bond, and you gain a hydrogen. And so that is what's happening when you're reducing NAD+ into NADH. Okay, so all those coenzymes that we just reduced into NADH and FADH2 are going to come to the mitochondrial matrix, and we are going to enter the electron transport chain. This is where most of your ATP is going to come from. And so the way it works is that NADH and FADH2 are going to come in, and they're going to donate their electrons to these electron carriers. So that means that NADH and FADH2 are oxidized because they're donating their electrons, thereby becoming NAD+ and FAD. But that also means that these electron carriers are being reduced because they are receiving those electrons. Okay? And now those electrons are going to be passed down the electron transport chain, and they're going to move from a high energy state to a low energy state, because as they're going down the chain, they are releasing energy, and that energy is used to pump hydrogen ions into that intermembrane space. And that establishes an electrochemical gradient called chemiosmosis. Okay? Now, along the chain, the electron is going to be accepted by oxygen. Remember, oxygen is the final electron acceptor along the electron transport chain. The oxygen is then going to split, and it's going to form a bond with hydrogen ions to form a molecule of water, right? So that's why water is a product of cellular respiration. It is a waste product of cellular respiration. But all the hydrogen ions in the intermembrane space, right, they've created this electrochemical gradient, and now you also have a higher concentration of those hydrogen ions in the intermembrane space as opposed to the mitochondrial matrix. And what happens when a molecule wants to move by diffusion? Diffusion means that that molecule moves from an area of high concentration to an area of low concentration. Those hydrogen ions want to diffuse to the other side of the membrane, but they can't do it, right? Because they're polar molecules. They are not lipid soluble. They're not going to be able to go through the plasma membrane. And so they need to be taken through the ATP synthase, right? So they're diffusing through the ATP synthase, and for each hydrogen ion, you're getting a certain amount of energy that is being used to bond a phosphate to an adenine diphosphate, creating an adenosine triphosphate molecule. Okay, and it's this process, this oxidative phosphorylation, that results in the majority of the ATP in the entirety of the cellular respiration process. Now again, you want to keep count of the products in each step of the aerobic reaction, because what we just went over, all of it is aerobic, except for glycolysis, that is anaerobic. But Krebs cycle and the electron transport chain require oxygen to occur. Okay, they are aerobic. So here, you can see that our direct products of glycolysis are 2 ATP and 2 NADH. And again, that NADH is going to go to the electron transport chain, and ultimately it's going to result in the production of 6 ATP. Okay? And glycolysis, again, directly produces 2 ATP. That is why it's called substrate level phosphorylation. You then have the pyruvate oxidation producing 2 NADH, for an ultimate yield of 6 ATP at the electron transport chain. The Krebs cycle directly produces 2 ATP. It also produces 6 NADH for an ultimate ATP yield of about 18 at the electron transport chain, and then it produces 2 FADH2 for the ultimate yield of about 4 ATP. So under best case scenarios, if this process was to work flawlessly, you would result in about 38 ATP. Now that's not always the case. Normally you only get about 30 to 32, but best case scenario, you wind up with 38, okay? Now remember, these yields of ATP are for 1 molecule of glucose. So next, we want to go over what happens if oxygen is absent through the entirety of the process. If oxygen is absent, then we undergo a process called fermentation. Fermentation is, again, an anaerobic process that occurs without oxygen. It's not a very efficient process, but it still produces ATP. It's just it only produces 2 ATP as opposed to 38 ATP. So it's not an efficient process by any means. There are 2 types of fermentation. There's alcoholic and lactic acid. And for both of these types of fermentation, the first step is always glycolysis because you need the glucose to be split into the pyruvate molecules. Okay? And this all occurs in the cytoplasm. So again, you have 2 types of fermentation. You have lactic acid fermentation and alcoholic fermentation. The way it works is just a little bit different. There's a few different intermediary steps, but you guys want to ultimately be familiar with the products. So lactic acid occurs in the muscles. When you've been working out very heavily, you run out of oxygen and your muscles start to undergo lactic acid fermentation. Lactic acid fermentation doesn't have any electron transport system. It just has NADH molecules transferring electrons directly to pyruvate. Okay? It's transferring the electrons directly to pyruvate. So you can see here in this picture that as you're splitting glucose, once again into 2 pyruvate molecules, you get the production of 2 ATP again, net production of ATP. You invest 2 ATP and you get the net production of 2 ATP, and you also get the production of 2 NADH, so 1 NADH per pyruvate molecule. Those NADHs come back, right, and they transfer their electrons to pyruvate, so they're reducing pyruvate to form lactic acid as a byproduct. So the products of lactic acid fermentation are 2 ATP directly and 2 lactic acids, 1 lactic acid per pyruvate. Alcohol fermentation is a little different. It's not too different. It still has that NAD plus regeneration, right? Because the NADH is going to donate its electrons to a carbon compound. So here we have glucose again. We have the net production of 2 ATP again. We have the production of 2 NADH and the split of the glucose into the pyruvate molecules. But the pyruvate undergoes another change in form that's going to result in the release of carbon dioxide and the production of what's called acetaldehyde. So acetaldehyde, it produces 1 acetaldehyde per pyruvate. And so the NADH then donates their electrons to this derivative of pyruvate called acetaldehyde, and that produces ethanol. Okay, so the products of alcohol fermentation are 2 carbon dioxide, 2 ATP, and 2 ethanol. That is fermentation in a nutshell. So ultimately, fermentation gives us many different things that we use in our daily lives. Lactic acid fermentation gives us soy sauce, cheese, and yogurt. Alcohol fermentation gives us beer, wine, and bread. So this is a quick overview, and you want to be able to sit here without looking at this, and you want to be able to remake this chart all by yourself. Okay. So in aerobic respiration, our reactants are glucose and oxygen. In anaerobic respiration, our reactant is just glucose because it occurs in the absence of oxygen. That's why it's anaerobic. Our products in aerobic respiration are ATP, water, and carbon dioxide. The carbon dioxide is a product of the Krebs cycle. The water is a product of the electron transport chain. In anaerobic respiration, depending on the type of fermentation that takes place, your product is going to always be 2 ATP, but in lactic acid fermentation, you're also going to get the product of 2 lactic acid. If it's alcohol fermentation, you're also going to get ethanol and 2 carbon dioxides. So make sure you know your different products per the different types of fermentation. The location is a little different again. Aerobic takes place in the cytoplasm for glycolysis, but primarily the mitochondria. Anaerobic takes place purely in the cytoplasm. There's different stages per each. Aerobic has glycolysis, Krebs cycle, and oxidative phosphorylation. Anaerobic has glycolysis and fermentation. And then, of course, aerobic is so much more efficient than anaerobic because you get the production of 38 ATP, and in anaerobic you only get the production of 2. This is just an overview of some of the vocabulary. Cellular respiration, again, is just a set of metabolic reactions and processes that take place in the cells of organisms to convert biochemical energy from nutrients into adenosine triphosphate, or ATP. Aerobic respiration is the process of producing cellular energy involving oxygen. So again, both oxygen and glucose are your reactants. In anaerobic respiration, this is respiration without oxygen, so only glucose is the reactant. Substrate level phosphorylation is the metabolic reaction that directly results in the formation of ATP by the direct transfer of a phosphoryl group to ADP. Oxidative phosphorylation is the process in which ATP is formed as a result of the transfer of electrons from NADH or FADH2 to oxygen by a series of electron carriers. And then it is ATP synthase that is going to transfer that phosphate to the ADP to form ATP. Again, a redox reaction is just a chemical reaction that involves the transfer of electrons between 2 chemical species. And what we mean by that is that oxygen and hydrogen are 2 different species. Oxygen and carbon are 2 different species. And so when you get the transfer of electron between 2 different species, that is a redox reaction. And that is it for this video. I do hope it helps. Let me know if you have any questions, and good luck on any exam.